Okay, so in this last section, this last lecture for Chapter 8, we're going to be looking at what is referred to as the Calvin cycle. The Calvin cycle is where we're going to see carbon dioxides are going to be recombined with other pre-existing molecules to give you sugars. Now, everything we talked about the light reactions, as you see up here in this figure, all the light reactions are centered around and in the thylakoid discs. Calvin cycle is going to be occurring in the stroma. Now, just like we saw with the citric acid cycle, here at the Calvin cycle, since it is a cycle, there is no beginning, no ending. Things feed in, things come out, but there's no beginning, no ending. It's a constant churning of enzymes, substrates, products, intermediaries getting fed in, intermediaries getting taken out for other things. But what links the light reaction to the Calvin cycle is the fact that all the energy to drive the Calvin cycle comes from the ATP and the NADPH that are generated through photosynthesis in the light reactions. Now, when it comes to the Calvin cycle, what I expect you to know, three things: fixation, reduction, regeneration. The three stages of the Calvin cycle. Now, when it comes to specifics, the who, the what enzyme, blah, 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 no, I don't expect you to know that. If you really want to know that, you really want to study that, take plant biology with Dr. Neely. What we're going to see is the fixation stage, stage 1, is going to be the fixation of carbon dioxide to a pre-existing molecule known as RUBP. RUBP is going to combine with CO2. It's going to be reconfigured into different molecules. Those molecules are then going to go through what is known as the reduction stage, stage 2. Stage 2, energy from ATP and ATPH is going to be added in so that everything can be recombined, reconfigured to give you GA3P, or in other textbooks it's referred to as G3P. G3P, glyceraldehyde 3-phosphate. Remember, that's the midpoint in glycolysis when glucose is split in half, giving you the two parts, two halves. G3P is what gets reconfigured to form pyruvate. Well, here you go. We've now just made half a glucose. We're going to see half that glucose is going to get kicked out, and the remaining molecules are then going to go through stage 3, regeneration, where they're recombined, reconfigured to generate RUBP again. So when you look at it, incoming CO2 molecules. CO2 molecules are going to combine with RUBP. 3 CO2s, 3 RUBPs. And if you count, 1, 2, 3, 4, 5 carbons in RUBP, plus 1 coming from CO2. So that means for every Rubisco that's being made, that is then going to go through the remainder of the carbon fixation into the reduction stages. 3 6-carbon molecules. So that means a total of 18 carbons. Through the carbon fixation into the reduction stage, those 3 molecules are going to get split and recombined to form 6. 3 PGAs. 6 times 3 is still 18. All the carbons are there. They're just now looking different. These 6 molecules are going to have ATP. They're going to be phosphorylated from ATP. And then they're going to be reduced from NADPH. And you're going to get 6 3-carbon molecules known as Ga3P, glyceraldehyde 3-phosphate, half a glucose. One of those is going to get kicked out. It'll combine later with another one to form glucose or fructose or whatever the plant needs. The remaining 5 molecules, though, can't do anything with. So what the stage 3 is going to do as we go through this is that Ga3P is going to get turned back into RUBP. 5 Ga3Ps, 5 times 3, 15. Going to take some energy to recombine, reconfigure, make new shapes, and you're going to end up with 3 5-carbons of RUBP. The numbers always have to equal the same. 5 plus 1 gives you 6. 6 times 3 is 18. 6 times 3 here in this stage, still 18. 6 times 3 at this stage still gives you 18. 1's lost. 5 times 3 is 15. And then 5 times 3 is 15 again. It all adds up. It all remains the same. Okay? And that's basically the level I want you to know it at. Calvin cycle, 3 stages. What's happening in a sentence or two for stage 1? What's happening in a sentence or two for stage 2? What's happening stage 3? Hide the specifics, take plant biology. See here, another link for a video that will help explain the process a little bit more, give you an animation to see it. Again, as always, I will embed some videos in the Canvas page for Chapter 8.